Immunology Insights, Basics and Beyond, Chapter 26, Types of Vaccines. Marcello here. Welcome back to Immunology Insights, Basics and Beyond. In our last chapter, we explored how vaccines work, how they stimulate the immune system to build long-lasting protection without ever causing the disease itself. But as we hinted, not all vaccines work the same way. Their composition and delivery methods vary depending on the nature of the pathogen, the immune response needed, and the population being vaccinated. Today, we will dive deeper into the different types of vaccines, how they are developed, and how each is strategically chosen to prevent specific infectious diseases around the world. So let's talk science. Let's look at the different types of vaccines. There isn't just one kind of vaccine. There are several, each using a different approach to teach the immune system what to look for. These differences matter. Some vaccines aim to generate robust, lifelong immunity with just a single dose, while others need to be boosted periodically. Some are better suited to emergency outbreaks, others to long-term disease elimination strategies. So let's have a closer look. First, we have the live attenuated vaccines. These vaccines contain a weakened but still living version of the pathogen. Because the microbe can still replicate, albeit very slowly, it mimics a natural infection and stimulates a strong, long-lasting immune response, often with just one or two doses. To make a pathogen less harmful while keeping it immunogenic, scientists grow it under unusual conditions, such as in non-human cells or at cooler temperatures, over many generations. This process leads to mutations that weakens its ability to cause diseases in humans, but not its ability to trigger a strong immune response. Examples here are the measles, mumps and rubella MMR vaccines, the chickenpox vaccines, or the yellow fever vaccines. Pros are here that live attenuated vaccines elicit a strong and broad immune response, often with just one dose. They offer long-lasting protection, often lifelong, and they closely mimic a natural infection. Of course, there are also cons. They are not suitable for immunocompromised individuals. They require a cold chain storage and careful handling, and there is a slight risk of reversion to a more virulent form through extremely rare. Second, we have the inactivated or killed vaccines. These use pathogens that have been completely inactivated, usually by heat, chemicals or radiation, so they cannot replicate at all. They are very safe and cannot cause the disease. Here the examples are polio, hepatitis I, or rabies vaccines. Pros are that they are very safe even for people with weakened immune systems. They are stable in storage and easier to handle than live vaccines. Cons are that they often require booster doses to maintain protection, and they may generate a weaker immune response than live vaccines. Third, we have subunit, recombinant or conjugate vaccines. These vaccines include only parts of the pathogen, such as proteins or sugars, often chosen for their ability to trigger a strong immune response. They do not contain whole pathogens, making them very safe. Examples here are the vaccines against human papillomavirus, hepatitis B, and pneumococcal vaccines. The pros are that they have fewer side effects, as only essential antigens are used. They are safe for all populations, including immunocompromised people. And they are highly targeted, reducing unnecessary immune activation. Cons are they often require multiple doses or boosters, and they may need adjuvants to strengthen the immune response. Fourth, we have the toxoid vaccines, used when a bacterium causes illness through toxins and not the direct infection. These vaccines contain inactivated toxins called toxoids, which train the immune system to neutralize the toxin. Examples here are the tetanus and diphtheria vaccines. Pros are that they are highly effective against toxin-mediated diseases, and they are of course very safe and stable. The cons are that they do not protect against the bacterium itself, only the toxin. and there are boosters needed to maintain protection. Fifth, we have the mRNA vaccines. These are the newcomers in the vaccine world, and they have made a huge impact in a short time. Rather than delivering a protein or weakened pathogen, mRNA vaccines contain genetic instructions, specifically messenger RNA, so mRNA, that tells our own cells to produce a harmless piece of the virus, such as the spike protein of SARS-CoV-2. Once the protein is made, the immune system recognizes it as foreign and mounts a defense, creating antibodies and memory cells in the process. The idea of using mRNA as a therapeutic tool had been explored for decades, but the technology only recently matured enough to be safe and effective for large-scale use. The COVID-19 pandemic accelerated development, leading to the emergency approval of the Pfizer-BioNTech and Moderna vaccines, both of which showed remarkably high efficacy in clinical trials. One of the greatest strengths of mRNA vaccines is their flexibility. Scientists can quickly redesign the mRNA code to match the new virus variants, making it an ideal platform for rapid response vaccines for future pandemics. The production process of mRNA vaccines is also faster and more scalable than traditional methods, since it doesn't require growing pathogens in eggs or cell culture. It is also important to note that mRNA vaccines are very safe. The mRNA is transient, meaning it breaks down naturally in the body and does not integrate into your DNA. The viral protein it encodes is only produced briefly, but the immune response it triggers is long-lasting. The fear of mRNA being long-lasting or integrating into the host DNA is not substantiated because it goes against the very nature of mRNA. But it doesn't stop at infectious diseases. The same mRNA technology is being explored for personalized cancer vaccines. In this approach, scientists identify unique mutations in a patient's tumor and design the mRNA that instructs cells to produce tumor-specific antigens. This could help the immune system recognize and destroy cancer cells, something it often struggles to do on its own. An example for mRNA vaccines is of course the Pfizer-BioNTech and Moderna COVID-19 vaccines. Pros are that it's rapid to design and produce, ideal for fast-evolving viruses. It induces a strong immune response, including both antibodies and T cells. There is of course no live virus involved, eliminating the risk of infection. And it holds a promise for also non-infectious diseases, including cancer. Cons are that mRNA vaccines require ultra-cold storage or cold storage depending on the formulation. They may cause short-term side effects like fatigue or mild fever, and they are still relatively new, with long-term data continuing to accumulate. 6. We have the viral vector vaccines. They use a harmless virus or vector to deliver genetic instructions into the cells, causing them to produce an antigen from the target pathogen. Examples here are AstraZeneca's and Johnson & Johnson's COVID-19 vaccines. Pros are that they also cause a strong immune response, including cellular immunity. They are also a flexible platform for rapid adaptation to new diseases. Cons are that some people may have pre-existing immunity to the vector virus itself, which can reduce effectiveness, and the storage needs vary depending on the vector used. 7th, passive immunization, which are added only for the sake of completeness. In contrast to vaccines, which train the immune system over time, passive immunization provides immediate protection by delivering ready-made antibodies. This approach is used in emergencies, such as the exposure to rabies, where antibodies are given alongside the vaccines. Pros are here a rapid, short-term protection. The cons, of course, that no immune memory is formed and protection fades away within weeks. It is important to choose the right vaccine for the right disease. Each vaccine type has its own strengths and weaknesses, making them more or less suitable depending on the pathogen, the population, and the goal of the vaccination campaign. For example, in areas with limited healthcare access, a vaccine that requires fewer doses and can be stored without refrigeration is ideal. For diseases like measles, where strong and durable immunity is crucial, a live attenuated vaccine is preferred. For diseases that change rapidly, like influenza or coronaviruses, platforms like mRNA or viral vectors allow rapid updates and deployment. Let's have a quick look at future directions in vaccine development. Vaccine science continues to evolve, and several cutting-edge innovations are on the horizon to be hopefully developed soon. First, a universal flu vaccine, aiming to provide long-term protection across multiple flu strains without annual reformulation. Second, cancer vaccines. Therapeutic vaccines are being explored to help the immune system to recognize and attack tumors. Third, personalized vaccines. Advances in genomic medicine may lead to vaccines tailored to an individual immune profile, improving both efficacy and safety. Fourth, needle-free delivery. New delivery systems like nasal sprays, oral capsules, and skin patches could simplify vaccination and increase compliance, especially in mass campaigns. To conclude, understanding the different types of vaccines helps us appreciate just how strategic and sophisticated our fight against disease has become. Each vaccine type is a carefully engineered solution, optimized for specific pathogens, populations, and public health goals. From classic live vaccines to cutting-edge mRNA platforms, the diversity of vaccine technologies is one of the great triumphs of modern medicine. In the next chapter, we will take a closer look at herd immunity, explaining how widespread vaccination doesn't just protect individuals. It protects entire communities, including those who can't be vaccinated themselves. Before closing, I have a small request. If you enjoyed my content and want to hear more, like and subscribe. It's a small step for you, but it helps me a lot. So see you next time, and thank you for listening.